Talo Falava, and welcome to another additional episode of our curriculum support program, Bilang Soa Om. Special Fatalofa to all our intended audience, of course the Agricultural Science Year 10 and Year 11 students, Talofa Tamamatini. Today, our lesson will be based on our curriculum textbook, Strand 1, which is agriculture. Our units we will be learning today about Punnett square, phenotype, genotype. We will be learning about the crosses of monohybrid and dihybrid. And of course, hopefully by the end of the lesson, we will be able to solve any problems relating to monohybrid and dihybrid crosses, understanding genotype and phenotype using the genetic chart or the Punnett square. But before we go on to our lesson, I would just like to remind you that you can always view our lessons and our program as many times as you want on our MESC Online Learning Facebook page. Enjoy learning agriculture science.
Hi, it's Mr. Anderson, and today I'm going to give you a beginner's guide to Punnett squares. There are a few mistakes that students always make when they're doing Punnett squares, so I hope to kind of clear those up. First of all, we should talk about the namesake. This is Reginald Punnett. He didn't really work with Punnett squares, however, he did work with genetics quite a bit. He did work with mimicry in the butterflies, and so his name is probably associated with genetics just as much as Mendel. And it's through the use of the Punnett square. Now, the Punnett square is often overused as just a quick way to solve genetics problems without really understanding what's going on with the genetics. And so I want to kind of get to that root. And if you could remember one thing from this whole video podcast, it's this right here. The two sides of a Punnett square represent the alternatives after meiosis. In other words, you have a bunch of genes and you give half of those genes to a sperm or an egg, and that happens through meiosis. And so the organization of those gametes, this case it's just a monohybrid cross, are going to be on either side of this, just like a flip of the coin. This would be for one parent, and then this would be the other parent on the other side. And so what do the boxes on a Punnett square stand for? They simply stand for all the alternatives that could occur if we had mating between each of these different gametes. And so let's get to some examples, and hopefully that'll help. So we're going to start with a monohybrid cross. A monohybrid cross is simply a cross that is looking at one trait. And so let's do one that's really, really simple. And so let's say we're crossing purple flowers that are homozygous purple with those that are homozygous white flowers. In other words, this is the dominant trait, this is the recessive trait. And so if you look at the parents, what you want to do first of all is figure out what are the possible gametes that could be produced in meiosis. In other words, this one could either give a big P or it could give a big P. What does that mean? It can only give one thing. It can only give a big P, or that dominant allele. And so if you're doing a problem like this, you don't even need a big Punnett square. One parent can only contribute big P. So let's look at the other parent. The other parent can either give a little p, and I try to make them really small, or a little p, because p's look the same. And so the other parent can only give a little p, and so we could put that on the other side. And so what are the opportunities that we could have as far as fertilization goes? Well, this one's automatically going to give a big P. The other one's going to give a little p. And so this is the only possible outcome we could get between a cross of a homozygous dominant and a homozygous recessive. And so you don't really need a big Punnett square. Now you could do that, you could fill it in, big P over here, little p over here. But if you do that, you're going to get the same thing in all of the boxes. And so it's still a 1-to-1 ratio. In other words, 100% of the time you're going to have that. Okay, so let's get to one that's a little more complicated. Let's say we have a heterozygous cross. And so this is for purple flowers as well. Well, if you look at this one, now the problem's changed a little bit. This one could give a big P, but it also could give a little p. And so we have to show both of those possible gametes of meiosis. And so this would be the big P, and then this would be the little p over here. So half of the time it's going to give the big P, half of the time it's going to give the little p. If we look at the other parent, same thing. It's going to give the big P half of the time, and it's going to give the little p half of the time as well. So we're going to put that here. Now we simply fill in the boxes. And so this would be a big P with a big P because I'm taking this here and that there. This is going to go over to here to give us a big P and a little p. By convention, we usually write the dominant allele first. So that would be one alternative here. Here would be a big P, little p as well. So we get the big P here and the little p here. And then finally, we're going to get little p over here and a little p over here, since they're each contributing a little p. And so what do we get from this cross? Well, we get a 1 to 2, since these are exactly the same, to 1 genotypic ratio, because the genotype is the letter. So there's one that's like this, there's two that look like this, and there's one that looks like that. So that's going to be our 1 to 2 to 1 genotypic ratio. What about phenotypic? Well, this one's going to be a purple flower, and so are these other two. And so if we're looking at the phenotypic ratio now is going to be a 3 to 1 ratio. We're going to have 3 purple to every 1 white that we have. Okay, let's try another one. Let's say we're looking at incomplete dominance. So incomplete dominance, a snapdragon would be an example of that. A snapdragon has 2 genes. If it has a red gene and a white gene, then it's going to be pink. And so this one actually has 2 alleles that it can contribute, and the same on the other side. And so we just write those out. So this would be the parent. This would be a 50% chance of giving the red, 50% chance of the white. And then the same thing on this side over here. So now if we fill in our Punnett square, like that, what do we get for all the different choices? Well, now we have a 1 to 2 to 1 genotypic ratio, but we also have a 1 to 2 to 1 phenotypic ratio. So if you're doing a question where it's incomplete dominance, you use a Punnett square the same way. Codominance would be the same way. The difference is in incomplete dominance, the heterozygous or the hybrids are going to be somewhere between the two. If it's codominance, they're going to express both of those genes, both of those proteins. And now let's try one that's a sex-linked chromosome or an X-linked chromosome. And so in this one, we've got a parent. So this is a mom because she's XX chromosome, and she's a carrier of, let's say, color blindness, the gene. And this is a dad that's normal. And so I'm going to put dad up here, X, Y, because half of the time he's going to give the X and half of the time he's going to give the Y. If we put mom over here, I'm going to put that carrier up there. She is not colorblind because she has one deficient gene, colorblind gene, but she has another gene that works well on her other X chromosome. So if I fill in this one here, it's going to be XCX. So this would be a female because 2 X chromosomes, but they're going to be a carrier of that gene. If we look down here, this would just be a normal female. If we look down here, I'm grabbing the X from here and the Y from up there, so that'd be XY, so that'd be a normal male. And then if we look at this one right here, this would be a male who's colorblind. The reason he's colorblind is that he doesn't have an X chromosome or another gene as a backup copy to that. So those are monohybrid crosses, and usually students do fairly well on those. Next are dihybrid crosses, and this is where the mistake really starts. And so if we look at this parent, this is a typical dihybrid cross. Let me tell you what the letters stand for. The R stands for round pea seeds, and the Y stands for yellow seeds. If it's the recessive, that stands for wrinkled, and if it's a little y, that stands for green. And so we're looking at a dihybrid, so that means 2 traits. We're looking at seed shape, round or wrinkled, and seed color, yellow or green. So now we have to do a dihybrid cross. And so the tendency, if we look at this parent, the tendency is to see that there are 4 letters over here, there's 4 boxes over here, and then you just simply write them out. Big R, big Y, little y, and then you get a weird answer and you don't know what to do with it, okay? That's wrong. That's a mistake, okay? Whenever you're figuring out the gametes, remember that you have to give one of each letter. In other words, each of the gametes is going to have one of each of the alleles. And so let me clear this mess out of the way. So what do? Let's say this parent right here. And I'm going to write it up here so it makes a little more sense. Big R, big Y, little y. What possibilities could they produce? So they're giving one of each letter, remember. Well, they could give the big R and the big Y. So big R, big Y. That'd be one possibility. They could also give the big R and the little y. So they could give the big R, little y. They could also give the little r, big Y, or the little r, little y. Since they're giving one of each color, there's only one of each letter, there's only 4 possibilities that they can give. So it's getting to be kind of a mess, but those are the 4 right here. And so those 4 are going to go across the top. So we've got big R, big Y, big R, little y, little r, big Y, little r, little y. So those are the 4 gametes that you could produce. In other words, with this parent, you can only get 4 combinations of each of the 2 letters. Same thing down this side. So it's going to be same thing on written down this side because this other parent is going to be exactly the same. Okay, so it would take me a long time to write all of those out. So let me throw those in here. So these would be the parents, all the possible gametes you could get. And if I fill those in, these first 9, if you look at them, let me get a color that's different. Let me grab a yellow color. And so this one right here is going to be round and it's going to be yellow. So it's going to be round and yellow. And this one you can see it's going to be round and yellow and round and yellow and round and yellow. And yellow and round and yellow and round and yellow and round and yellow and round and yellow. In other words, 9 of them are going to be round and yellow in shape. And the only reason why is that the R is dominant, and so you could have one like this where they're hybrid for both and they're still going to be round and yellow. And so let me try to add the next ones. So what about these next ones? Well, the next ones are going to be round and green. So let me get a different color. So these ones are going to be round and green because the round is dominant, but they don't have the dominant for the yellow, so they're not going to be. Let me get rid of that. Let's add the next ones. So these ones are going to be wrinkled and yellow. So again, I got to get a different pen. So these ones are going to be wrinkled and yellow. And then if we look at the last one, it's going to get all of the recessive alleles. And so that one's going to be getting green. It's going to be green and wrinkled. Okay, and so when we say there's a 9:3 to 3:1 ratio, that's just a typical dihybrid cross. We're going to have 9 of the phenotypes that are round and yellow, 3 of the phenotypes that are round and green, 3 of the phenotypes that are yellow and wrinkled, and then only 1 of the ones that's not. And that's only going to work if you are able to set up your gametes correctly on the side. Now it's super hard for you to answer a question like this on a test. You're rarely going to have to draw a dihybrid cross, but it's important that you understand the concept of it because most of the genes inside your body are not caused by one gene, they're caused by multiple genes. So how tall you are is caused by probably a dozen different genes inside your body. So you can imagine how big the Punnett square is going to be for that. An example of that I'll leave you with would be this one. So let's say we have a parent here, and the parent is big R, big Y, little y, little r, little r, little y, little y. The question I'm asking you is, how big would your Punnett square have to be? And so you're going to have to figure out what are all the possible gametes that you could get from both of those, and then draw it out, figure out all the possibilities you can get. If you're thinking it's going to be a 4x4, you're doing way too much work. And so those are Punnett squares, and I hope that's helpful. Hi, it's Mr. Anderson, and welcome to Biology Essentials video 29. This is on Mendelian genetics, and it's called Mendelian genetics. It's named after Gregor Mendel. In biology, there are 2 famous names, Darwin, who was famous and controversial in his own time, and Gregor Mendel, who died in obscurity. But both of them made huge advances in the field of biology, and Gregor Mendel in the area of genetics. If they could have gotten together, those 2 theories, when they finally came together as modern synthesis, it was really powerful. Unfortunately, he dies in obscurity. But this is what he did. He was crossing pea plants, and so he can take one pea plant and you use a paintbrush to transfer pollen from one to another so you know who are the parents. He would then create offspring as a result of that. Now, peas are great because they have a number of different characteristics, and more importantly, you can make a lot of them really quickly. So in a pod, each of these peas is an actual new organism. So you can plant those and see how they grow. And so he figured out a lot of genetics as a result of that. And so genetics in this podcast I'll talk about are Mendelian or simple genetics. He identified the gene and he came up with two laws, the law of segregation and independent assortment. I'll talk about those. We'll also have some problems, so I'll have some practice problems that you can try and I'll show you how to work those out. And we'll finish with genetic disorders. The example I'll talk about is Huntington's disease. And with genetic testing, it opens up this whole idea of ethics and privacy. Now, what won't I be talking about? I won't be talking about linked genes. In other words, if genes are ever on the same chromosome or on a sex chromosome or caused by multi-genes, it gets really complex. And so I'll talk about those in the next podcast on advanced genetics. But know this, that simple Mendelian genetics, the rules are simple rules of math, rules of probability. And if you understand those and how to do the first cross that I show you, you should do well in most any of the crosses you get in genetics. And so this is what Mendel did. He crossed purple flowers with white flowers, and he got purple flowers. Now, a few things that you should know. The first cross in any genetic cross is called the P cross, or the parental cross. And then the offspring of that are called the F1, or the filial one, or the offspring of that cross. And so at the time of Mendel, everybody believed in this idea of blending, that if you cross two parents, so mom and dad, their kids look a lot like them, and so it's maybe a blending of they didn't know that they were genes, but something inside of them. And so when he crossed purple with white and got purple flowers, that made total sense back then. This kind of fits in this blending. But what he did next was he crossed these purple flowers with themselves. And what he got was a 3-to-1 ratio of purple to white. And that white returned just as crystal white as that first white was to begin with. And so what he said is that there's a character, a trait that's passed through here, but we now identify that as a gene that's carried distinctly through each of those generations and then shows up again. Now, if you know anything about genetics, you'd know that in simple Mendelian genetics, it looks like purple is dominant, white is recessive, but we know that because of the work of Mendel. And so this is what the second cross is, the one that was puzzling, and this is what he eventually figured out. So these offspring right here were hybrid for the trait. And so if we look at the parents would be big P, that represents one purple flower. Little p represents the white flower, and so each of these would be big P, little p. And so you can use a Punnett square. A Punnett square, you put the pollen or the male here, the female right here, and each of these genes get a certain column. And so this would be one parent, big P, little p, and so you simply write that across. So we got big P and little p here. We got big P and little p here, so this represents the two different parents crossing with each other. And then we simply figure out what comes. So here's big P, big P. So we get a big P from one, a big P from the other, so that'd be big P, big P. Since big P is dominant, we get a purple flower. On the next one, I'm going to take a big P from here, a little p from here, and so that would be still purple because this one is a dominant allele or a dominant gene. Down here, we get a big P from here and a little p from here. And so that's purple. And the reason we get white flowers is that you get a little p from both of the parents. And so the neat thing about a Punnett square, and that's what this is it not only allows you to quickly do the probability, but it shows you the percent we should get in those offspring. In other words, 3 of them should be purple, and 1 of them should be white. So we should have a 3-to-1 ratio. And if you ever get a cross like this in a problem, for example, a cross like this, just if you're not sure what the offspring are going to be, just do a simple Punnett square where these two alleles or versions of the gene are going to be on the top and these two are going to be on the side. And we'll do some practice problems in just a second. And so what are Mendel's laws? Well, the two things that he figured out are the law 1, Mendel's law 1, it's called the law of segregation, and Mendel's law 2 is the law of independent assortment. And so let's start with law 1. And so if we ever, and I got a coin here because the actual way you get genes are almost like a coin flip. So if you think about a coin, it has heads on one side and tails on the other. When you flip a coin, what are the odds that you're going to get heads or tails? Well, it's a 1 in 2 probability that you'll get heads. Same thing with genes. And so if this is that F1 generation, and this shouldn't be B, this should be P. So we'd say this is big P, little p. What are the odds that the offspring are going to get a big P Well, it's a 1 and 2. What are the odds that they're going to get a little p? It's 1 and 2. And so that separation of those two alleles is called segregation. And so this idea of segregation says that there's a 50% chance you're going to get either of these genes. And so that's segregation. They separate, and it's just random chance. The next one is the law of independent assortment. Law of independent assortment says that this gene, the gene that causes, for example, hitchhiker's thumb, which is where your thumb actually bends back, and the gene that causes an attached earlobe, so right here I've got a free earlobe, those two traits don't affect each other. In other words, they sort independently. And so we can work problems without mixing these two together. They're going to not influence one another. Now, sometimes we'll find, for example, that some things do travel together. So you'll notice that people who have red hair also have freckles. And that's because those two genes are actually found on the same chromosome, and so they seem to travel together. And so we're not going to deal with linked genes again. We'll do that a little later. So independent assortment means that traits don't affect each other. And so what I'm going to do next is I'm going to leave this for a second. These are 6 problems that I'll work through. But if you want to work these, you could pause the video at this point, and then you could come back and start the video again and see me work through each of these. So I'll pause. All right, so let me go through these. Question 1. A coin is flipped 4 times, comes up heads each time. What is the probability that the next coin flip will come up heads? Well, everything that's happened in the past can't influence anything that's going to come in the future, and so it's a 1/2 probability that you'll get heads. In other words, you could have 10 kids, they could all be boys. What are the odds that the next one's going to be a girl? It's still a 1 in 2 probability. Let's look at the next one. And so we've got some things up here. Round peas are going to be big R, and wrinkled peas are going to be little r. Yellow will be big Y, and dominant is going to be, or green is going to be little y. Generally, whatever is the dominant trait, we give that the capital letter. In this case, round gets the big R, and yellow gets the big Y. And so question number 2, classify the following as heterozygous or homozygous. Heterozygous means you have different genes or different alleles. Homozygous means you have the same. And so this first one, big R, would be homozygous dominant. They have the same. This would be heterozygous, and we also sometimes refer to that as hybrid. The next one would be homozygous recessive, and the next one would be heterozygous yellow, homozygous for the round. So it's going to be heterozygous and then homozygous dominant on the next one. So that tells you the alleles that you have. Let's look at number 3. What's the phenotype of the following? Well, this right here is going to be the genotype. In other words, big Y, little y is going to be the genes that you have. What's going to be the phenotype? Well, that's physically what you look like. And so for the first one, this one right here, even though its genotype is big Y, little y, or it's heterozygous for that, its phenotype would be yellow. So this is going to be yellow. This one's going to be round. This one's going to be green, and this one here is going to be yellow round. Phenotype is physically what you look like. Let's look at the next one, number 4. What's the probability of this cross? So we have 2 round seeds producing wrinkled seeds. Well, like I said before, if you ever get one of these, it's a simple monohybrid cross. I would always do a Punnett square. And so we'd put big R and little r on one side of my Punnett square. Big R, little r on the other side of my Punnett square. So what are the odds that I'm going to get wrinkled seeds? Well, there's a little r here, a little r here. And so there would be a 1 in 4 probability that we'd have wrinkled seeds. Because this one's going to be round, this one's going to be round, this one's going to be round as well. And so again, if you ever get a simple cross like that, do a Punnett square. Let's look at the next one. What's the probability that this cross would produce green seeds? Well, I do the same thing again. Big Y, little y, crossed with little y, and we're looking for green seeds. Green seeds, remember, are going to be little y, and so I could get it here, I could get it here, and so that is a 2 in 4 or a 1 in 2 probability that we're going to get green seeds from that. And so even though you might think you're super smart, do a Punnett square. You're never going to miss the problem then. Now this is a problem that we'll sometimes get on the AP Bio test as well. If these parents are crossed together, what are the odds that you'd get that? Well, to do this one, you'd have to actually set up a pretty intense Punnett square. And so if you get one like this, don't do a 4x4 Punnett square. Just work each of them individually. And so what do I mean by that? Well, let's start on the R's. What are the odds that if you produce this and that, you could get that? So let's do an R's first. So big R, little r, crossed with big R, big R. What are the odds that we're going to get big R, little r? Well, neither of these. But this is a big R, little r, this is a big R, little r. And so the odds of these two parents producing these offspring is going to be a 1 in 2 probability. So I'm going to write that right over here underneath the 1 in 2 probability. Now let's work the Y's. So let me get a different color. So if we do the Y's, what are the odds that these two parents are going to produce that offspring? Well, let's do those together. So here are the parents, big Y, little y, crossed with big Y, little y. So this is going to be big Y, little y, little y, big Y, little y, big Y, little y. So what are the odds that we're going to produce again big Y, little y? Well, it's a 2 in 4 or a 1 in 2 probability. So I'm going to write 1 in 2 here. Okay, so now instead of doing this huge unwieldy 4x4 Punnett square, what I've done is I've almost got there. Because the odds of getting this are 1 in 2, the odds of getting this are 1 in 2. So what are the odds of getting both of those? I simply multiply those together. And it's a 1 in 4. In other words, what are the odds of rolling one or flipping the coin and getting heads? 1/2. What are the odds of flipping 2 heads in a row? It's a half times a half or a fourth. And so you can solve problems like this just using what's called the law of multiplication, when these two things have to happen.
This activity is used to test your understanding about our lesson. And what you need to do is use this information here on the top, this information here, to fill in the spaces in the box below. Here we go. These two letters make up the organism genotype, or a set of alleles. This is the meaning of genotype, set of alleles. Here, these are the alleles. 2 letters representing each parent, one from the mother and one from the father. The appearance of organism is called phenotype. A phenotype is the physical characteristics of a living thing, features of a living thing. What you can describe of a living thing. Oa ni mea te whaamtalai nei se meola. O le wingana o le upu phenotype. Here we use the color green and yellow. Next is pure or hybrid. Pure is when the 2 letters are same, like this one. 2 capital letters and 2 small letters, meaning there is no mixing of gene. Pure are more like this one and this one. But the capital letter and small letter is hybrid, meaning there is a mixing of gene. Afatasi, palangi tama, sa motina mola afatasi. Hybrid or afatasi. Homozygous, an organism that carries 2 identical alleles is called homozygous. Homo means same. Yeah, this is homo. This is another homozygous. If the alleles are different, it is heterozygous. This is heterozygous. Okay, let's start filling in the gaps. Phenotype. What is the phenotype of these 2 letters, capital letter G and another capital letter G? What is your answer? Yes, the answer is green. The reason is that capital letter G here, physical phenotypes of capital letter G is green. Second one, capital letter G and small letter g, phenotype is green. Reason is that there is an involvement of a capital letter here. Capital letter is dominant. That is why the phenotype is green. Third one, small letter g, another small letter g. Phenotype is yellow. Yes, yellow. You got this from the key up here. Small letter g is yellow. Next, pure or hybrid. 2 capital letter means there is no mixing of gene. Samotama, Samotina, Lono winga ole, Biwa. Selau pasenel Biwa Samoa. Next, capital letter G, small letter g, hybrid or Biwa, hybrid. Reason, there is involvement of gene here, mixing of gene. Samotama, Palangitina, Mole, Afkasi. Next, pure or hybrid, small letter g, small letter g. Your answer is, and my answer is, pure. The reason is that there's no mixing of gene. 100% pure Samoan or pure Palang. Next, homozygous or heterozygous, capital letter G, capital letter G. The answer is homozygous dominant. Reason, homozygous, same. 2 capital letter. Dominant is when there is an involvement of a capital letter 1 or 2 capital letter, always dominant. Here, G, green, dominant. Next, homozygous or heterozygous, capital letter G, small letter g. Answer is heterozygous dominant. Heterozygous because there are different capital letter G, small letter g, different. Dominant here because there is an involvement of a capital letter. The last one is small letter g, homozygous or heterozygous. Answer is homozygous recessive. Reason, same, small letter, recessive, 2 small letter. The only recessive here, 2 small letter yellow. Okay, that is our activity for today. Thank you for your cooperation and participation. Until we meet again, Tofasoi Fua.